Oh, here we go.
Here we go. Oh, disclaimer, disclaimer, disclaimer. Not all the hosts may parrot the same views.
It's okay.
It's okay. It's okay.
Working with the same facts, we can still have different opinions. As long as we're working with the same facts.
We're going to discuss things. We're good to go. So let's start this show in 3, 2. This is TWIS. This Week in Science, episode number 764, recorded on Wednesday, March 11th, 2020. What does pandemic mean? Hi there. I'm Dr. Kiki, and tonight on TWIS, we will fill your head with chlamydia, pandemics, and glowing animals. But first...
Disclaimer, disclaimer, disclaimer. Pandemics are nothing new. Actually, while common, they are a rather new phenomenon for humans because for most of human history, humans preferred not to hang out where large groups of other humans were hanging out. Our nomadic hunter-gathering, frequent foraging forebears didn't hang out in any one location long enough to catch a seasonal anything. If something really deadly came along, it killed maybe 10 or 20 people in a single group, and that was it. It went no further. Then at some point, we began returning to the same places year after year, then staying all year in the same location. Farming, animal domestication took over foraging, populations blossomed. Humans living closely with other humans and goats and cows and pigs and chickens and wild dogs and everyone pooping all the time and never far enough away from town or water supplies with greater and greater frequency. And less surprise, humans have been riddled with one form of communicable illness or another as the world faces a truly comprehensible threat in the form of a virus, one that is quite possibly going to infect every one of us eventually and is more deadly than the regular seasonal flu. Societies are choosing to commit suicide as a solution. Corona canceling conferences, music and social gatherings, viral vacation plans, political gatherings in school classes, pandemically postponing political rallies, public transportation, symptomatically surrendering stock markets and sporting events. One thing that won't be canceled, regardless of what goes on elsewhere in the world, This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Hey, co-hosts, welcome back.
Hey, so happy to be back.
I know, you were both gone for a week. I replaced you, decided not to replace you. You're back. We had fun last week. I hope that both of you did also.
Yes, definitely. Disneyland was kind of empty.
Oh, what a great time to go.
It was an interesting experience.
I would imagine that. Oh boy, oh boy, oh boy. I wonder why it was empty. Could be that it's not quite spring break yet. Could also be that, oh hey, there's officially a pandemic. We're going to talk about that a little bit in this episode. And additionally, I have stories about chlamydia in the ocean. Bunch of rocks found outside of Neptune. And pillow talk. I will. Yes. Justin, what did you bring?
I've got where seasonal flus come from. This is a 4-year-old study, but I think it's worth talking about because it has some interesting insight into what's going on now, as well as dark DNA.
Dark.
98% of our DNA apparently doesn't do anything? I didn't know that.
Oh, it doesn't reflect light. No, I'm kidding. It's dark.
And then an undetermined story that'll be found before this we get to the end.
Okay, fantastic. I'm always up for
I got a
mysteries,
couple. There's a couple
especially
that
scientific mysteries. Blair, what's in the animal corner?
Oh my goodness. It was quite a week for animal science. I have multiple stories in the animal corner all about crazy colored critters. And then at the end of the show, I have a quick little story about dumb humans.
Oh, that seems to be all the news these days. Oh, humans, what have we done dumb this time? Something. But you know something that you can do that's very smart? You can subscribe to This Week in Science on your favorite podcast platform, Apple Podcasts, Google's podcast platform, Spotify, Stitcher, Spreaker, Pandora, Radio.com, TuneIn, all the places we are there. Also on YouTube and Facebook. You can tell someone to subscribe as well, or you can just search for TWIS.org. That's our website, and there's some subscription stuff there. This Week in Science. You can find us just about everywhere. Let's get into the show. To get started, oh boy. Yes, I mentioned the pandemics, and yeah, that's pretty much what we're going to talk about tonight. At least what I'm going to start off my segment of the show with tonight as I'm moving windows around on my screen. There's the screen. That's what I want to do. I'm going to share my screen because there is a wonderful map that we love to share of the spread of coronavirus. I think we are starting to see the exponential uptick in cases globally as a result of the spread of this novel virus to humanity. The World Health Organization today, just today, finally called it a pandemic. And in honor of it being called a pandemic, first question is, what is a pandemic? Well, pretty much it means that there is sustained spread across multiple continents around the globe. Not just an epidemic, which is sustained spread in one area. The flu, seasonal flu, that's not even necessarily pandemic. Pandemics are when one particular viral strain starts spreading globally, has that sort of sustained spread. It potentially could have been argued that we reached pandemic stage over a week ago. However, many concerns go into making the declaration. But now that the declaration has been made, it means that financial resources and emergency health resources can be made available to countries around the world, which is good news. I can
see why they didn't want to, though, just because it does already, even without calling it that, it was causing some pretty intense behavior in all over, which I think is exactly what they were trying to avoid. Seems like maybe it didn't avoid that fully, but I understand where they're coming from because, you know, you had people buying out the shelves on bottled water, which in the Bay Area, I think I have my own issues with because like the taps still work. But anyway.
Totally fine.
Yeah, it's not like an earthquake that's going to affect municipal water supply. It's not, not that you're preparing for the wrong disaster by hoarding water.
Yeah.
Yes and no. So the argument goes that if it were to get extremely dire and people in the places that fix broken water lines are sick and cannot do that, potentially you're up against supply chain issues. And so you want to be ready for all eventualities. Most likely, the toilet paper will keep coming. The water will keep flowing. Everything's going to be fine. But for you too, and for you, our listener, I have a quiz. The COVID quiz.
Right.
Are you ready? Let's see how you do. Question number 1. COVID-19 is A, a kind of bird with a great memory. B, an acronym for the disease caused by the novel coronavirus discovered in China in 2019. C, a family of coronaviruses. Take your
I'm going to go with number 2.
Yeah, it's B.
It is B. That's right. COVID-19. The 19 stands for the year in which the novel coronavirus, the virus itself is known as SARS-CoV-2 now. It's a SARS coronavirus, it means that it's a respiratory coronavirus, and this is the second one of its kind that we have discovered. But COVID-19, that's the name that we've given the disease. So if you catch it, you don't just catch coronavirus, you catch COVID-19, because coronaviruses are a whole family of viruses. Question number 2. Which is better for reducing the probability of COVID-19 spread? Hand sanitizer or hand washing?
Hand washing.
Yeah.
Yay! Ding, 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 ding. Good job, co-hosts. Question number 3. Oh, actually, before I move on, do either of you know why hand washing is better?
It's the size of the virus. The soap and water for 20 seconds is more likely to displace it than just swishing it around with hand sanitizer.
Yeah, I thought it had to do with displacement, that like physically removing the virus is more important than deactivating it.
Yes. So there is that. And the reason the displacement happens is because the virus has a lipid bilayer. And the soap interacts with that lipid bilayer. And that's what makes it easy for the soap to pick it up and take it away. Yes. Little did you know the virus had a lipid bilayer. Number 3, speaking of sanitizing, which of the following is approved by the FDA for destroying SARS-CoV-2? Apple cider vinegar, colloidal silver, Clorox bleach, or none of the above?
Oh.
I'm going to go with, I like the apple cider one, but I'm going to go with none of the above.
Okay, and I'll take the vinegar, just since I'm not sure.
Justin gets it right. None of the above. Ding, 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 ding.
That's funny. I actually heard somebody tell somebody else vinegar today. I was like, I did not hear that before.
Nope. No. There has been nothing that has been tested and approved for actual sanitation against SARS-CoV-2 by the FDA. The coronavirus family, Clorox bleach and other bleach products have been shown to be useful against other coronaviruses, but nothing has actually been approved federally for use against SARS-CoV-2 at the public level. Dun, dun, dun. Little you know. But yeah, if you can use bleach, do that. If you can use soap and water, again, that can help lift it and take it away. Bleach could potentially destroy it. Alcohol, anything over 70% isopropyl alcohol will denature the virus after time. So the alcohol has to sit on the surface. You can't just put the alcohol on and then wipe it away with water. It has to sit on the surface to be able to be effective. All right. 4, face masks are best. A, filled with menthol at a rave. B, stockpiled in my basement with the toilet paper.
Boo.
Or C, used to protect healthcare professionals.
See?
Actually, I'm going to go with A on this one because from what I understand, this is not an airborne illness. So
living with a healthcare professional, I'm going to say C.
Yeah. Save the face masks for the professionals, people.
It's droplet. It's droplet communicable. So if somebody's coughing, or if somebody gets spit on you, then you can get it. So if you're a healthcare worker and you're up in somebody's business, that stuff can get on you.
Yeah. Or if you touch a surface that is contaminated and then touch your face, oopsie. So hand washing. Number 5, social distancing is the name of a punk band. B, a practice involving cancellation of public gatherings. C, a long distance relationship.
It's B and C.
What was the question?
Social
All right,
distancing. Yeah.
You're not gonna pick one, Justin?
I'll go with what was your answer, Blair? First one.
B and C.
Okay.
B.
I'll pick B.
Yeah.
I'll pick one of them.
Yes, social distancing is an effort by health agencies and governmental agencies to stop the spread of diseases, usually by telling people to not gather or by forcibly stopping the gathering of people. You can also choose self-isolation or quarantine, or you can choose a minimal social distance of like 6 feet away from people, because that seems to be the distance that the cloud of air droplets spreads when we it's the worst area to be in after a sneeze. Number 6, the average time to symptoms after infection is, and this really just came out recently, A, 2 days, B, 5 days, or C, 10 days.
This was the amount of time for what?
The time to symptoms.
Time to symptoms.
After you're infected, how long does it take for you to start showing symptoms?
Well, they had been saying 14 days until very recently.
Yeah, this is new, so I don't.
So I'm going to go with 10.
Yeah, I would go with 10.
5 days.
Really? Oh, that's good news, actually.
The average time now is 5 days, 5.1
Wow.
days.
That's really good news.
According to a recent estimate, yes. And finally...
Is it still 14 to clear it? It's 5 days to...
So this is actually very interesting. The symptoms, you seem to be... people with mild symptoms clear it and become less infectious sooner. And so they're thinking that they'll be able to send people home at 10 days after the start of their symptoms.
Okay.
If they've been in isolation or quarantine, that when after about 10 days of starting symptoms, if they don't.
So I think we should get half credit for that.
Yeah, half credit. Okay. And the final question, true or false?
True.
COVID-19 is just another flu.
False.
I already picked my answer. I got in too quick.
You picked your answer with truth. The answer is that's false. COVID-19 is not just another flu. It has completely different dynamics. We have no vaccine. We have no herd immunity of any kind. It's completely new, and it has a much higher mortality rate. So be safe out there, everyone. Wash your hands. Try not to touch your face. The minute you do, realize how hard it is not to touch. You have to sit on your hands all day long.
I realize I spend a lot of my day like this.
I work at home, so I figure I'm fine.
Yeah.
If I haven't been outside all day.
Child?
I wash bleach.
Have you
soap
watched
and water
your
and
child?
bleach dip.
No, not watched, but witnessed how they interact with the world. I've been watching my 6-year-old touch everything and then have fingers in mouth like immediately after. It's just,
Yeah.
Children are going to be the death of us all.
So someone in the chat room, this is a really good question. So someone in the chat room in YouTube is asking, what about touching mail? Which is a huge thing that is like all over. The people are worried about food that came from China.
Okay. Look, here's the thing. Think about that, but you should also think about the touch screen when you pay for anything, taking money out of an ATM machine, the electronic signature with your finger on everything. I mean, we're in a touchscreen society now. How in the world, there are things, there is a card that is very popular in Europe it's got that you just tap the card onto the thing and that pays. You don't have to insert it, push buttons, and do a thing. The card is present with you, boom, that's it. But most of the places you have to touch things. And then, you see people who are wearing gloves, who are handling like receipts and stuff. Those gloves that they're touching just touched 50 people's whatever it is. So now they're just protecting themselves, but now they're creating a vector point of their gloves for everybody else.
What I was gonna say is that mail is, as I understand it, is not a concern unless it's wet or has been wet within 24 hours. And so most mail takes at least 2 days to get places. And so knowing that, my understanding is your mail should be fine.
Okay, but that's what's weird about that's why this virus apparently is so communicable. Maybe it's this lipid layer on it, but it has been surviving in locations apparently much longer than that. Normally normally you're right. A virus lands on the surface 24 hours later, it's not active anymore. It's not going to infect. Apparently this strain of the coronavirus is persisting in the environment for a much longer period of time, and that's leading to its high transmissibility rates.
That's very interesting. Yeah.
So be ready for the mail. Don't coming in the mail. It's
my understanding is the mail is fine, but yes, wash your hands.
Wash your hands. You can use a glove. You can wear disposable gloves if you need to, but wash your hands. This is the easiest thing to do. And stop touching your face. I laugh as I say because I'm completely unable to do the same thing. Okay.
There's a video of a mayor somewhere reading the announcement from their public health department to asking people to stop touching their face. And then she's going, lick the finger, turn the page. Also, they go, da-da-da-da-da. So while she's live publicly telling people not to touch their face, she's licking her finger. It's most hilarious. It's very difficult. Yeah, we humans do this much more than we realize.
We do. Oh, humans, creatures of habit. Oh, but will we catch chlamydia from the Arctic Ocean?
Gosh, I hope not.
I mean, I'm not gonna.
Well, maybe, but more likely we'll just learn where chlamydia potentially came from and how animals became infected with this parasitic bacteria. We know it as a bacterium that is symbiotic in nature, needing other organisms to provide it with materials to survive. Researchers recently published in Current Biology their gene sequencing study doing a phylogenetic analysis of genes that they discovered when they sifted through the sediments from the bottom of the Arctic Ocean, the mid-Arctic ridge. They found not the bacteria themselves, because that didn't work so well. They haven't been able to get anything to grow in the lab either, for that matter, but they found a whole bunch of genes that fit within the phylogenetic description of the family of chlamydia. The Chlamydia bacteria, which really we haven't discovered anywhere in a non-symbiotic form. However, these genes indicate that chlamydia, or species related to the organisms we know as chlamydia, live in a very low oxygen, no oxygen environment under extremely high pressures, extremophiles. And they're living down there. We don't know how, because a lot of their genes seem to still be related to the ones we recognize for symbiosis. So perhaps there are other organisms down there in the mud and muck at the bottom of the ocean that chlamydia species are living off of. But that doesn't seem to, we haven't found anything that indicates which species, if any, those might be. Just the genes are there.
That's really fascinating.
Very fascinating.
I would check. I wonder if we got chlamydia from crabs. No. Well, I'm trying to think of what would be down at like sort of an Arctic-y seafloor that humans would ever have come into contact with. It's It's
Right.
puzzling.
How did they get started down there? Have they just ended up down there by this deep sea hydrothermal vent? Is like this vent field that's down there on the bottom of the ocean? This area, I love the name of it. They where they sampled the sediment is called Loki's Castle. So hospitable and tricky, I guess. Researchers in the study from Uppsala University in Sweden said finding chlamydia in this environment was completely unexpected and, of course, begged the question, what on earth were they doing there? And they still don't know. And so they got to figure it out. What are they doing down there? And they went on to say, by using genomic methods, we obtained a more clear image on the diversity of life. Every time we explore a different environment, we discover groups of microbes that are new to science. This just tells us how much is still left to discover. Yeah, and maybe chlamydia play a really big role ecologically. They were the most numerous or dominant species in terms of the genes that they discovered in their survey. So if they were so prominent in the ecosystem, based on the genes that they found, that means that they probably have a very influential role in the ecosystem. So what are they doing down there?
I know, but I'm not going near any mermaids anytime soon.
I mean, it just sounds like we live in an open system and nothing we have is unique. That's what I'm taking out of this.
Everything I can do, they can do better. Yeah.
Or similarly.
Or similarly. Exactly. Justin, tell me a story.
So this is normally This Week in Science. Of course, we are doing a story that happened this week in science. But this week I'm doing a story that happened in September of 2016. And it's when it was published anyway.
What?
Yeah. This was a computer model developed by scientists at the University of Chicago that shows small increases in transmission rates of the seasonal influenza A virus H3N2 can lead to rapid evolution of new strains that spread globally through human populations. So they published this back then in the Royal Society B, or Proceedings of the Royal Society B. And this was pretty interesting. So they showed that the seasonal flu largely originated from areas of East, South, and Southeast Asia, where population size and community dynamics increase transmission of endemic strains of the flu. This is quote voice of the lead author, assistant professor of ecology and evolution at the University of Chicago, Sarah Kobe. The transmissibility is a feature of the pathogen, but it's also a feature of the host population. So a host population that potentially has more crowding, larger classroom sizes for children, or even certain types of social contact networks potentially sustains higher transmission rates for the same virus or pathogen. There are 4 influenza strains that circulate in the human population. The viruses spread seasonally each year because of antigenic drift, which is they slightly change their genome. They don't become a completely new strain of the virus, but they evolve just enough so that we can still get sick from them. The H3N2 subtype causes the most disease each year. And genetic sequencing has shown that for 2000-2010, 87% of globally spreading strains of H3N2 originated in East, South, Southeast Asia. So this is pandemic. This is normal flu going pandemic and going everywhere in the world. It's the same flu, but it seems like it gets its start quite often in these Asian areas for some various reasons that they go into. So Colby and her colleague Frank Wen from the University of Chicago and Trevor Bedford of the Fred Hutchinson Cancer Research Center, they developed a computer model that took into account a lot of the conditions that they thought might be prevalent. The team used the model to test several hypotheses about why antigenetically new seasonal flu strains tend to emerge from the same part of the world, including seasonality. So 85% of Asia's population lives in tropical or subtropical regions with no distinct winter, which they say allows for continuous transmission of endemic flu strains, more opportunities for evolution of new strains because they don't have this sort of dormant phase, I suppose, from a cold weather type thing. Host population size, this area contains more than half of the world's population. That's many, many billions of people. So the sheer number of people contribute to the larger fraction of strains that originate and then spread globally. Host population turnover, they looked at, well, birth rates are higher because there's more people. Obviously, there's also more children. So they point the finger at children as being highly susceptible to catching the flu and then transmitting it amongst themselves, each other, and everybody else.
There's a reason kids are always sticky, right? It's because they're not great at washing their hands.
And the initial conditions are also interesting. So H3N2, that the most successful popular flu, first emerged in Hong Kong, 1968. Can you imagine they didn't have the seasonal flu before 1968? One hypothesis states that this gives the viral population an evolutionary head start because this one has been going for so long. It's had more opportunities. And so new epidemics, they think, will almost always begin in this area because of the amount of evolution that this strain has gotten. Also, transmission rates. Differences in transmission rates of a given virus or the expected number of secondary cases caused by a single infection can cause the evolution and affect the evolution of new strains. So they simulated some climate conditions, population dynamics, and patterns of flu epidemics in these regions, and they found that not only the transmission rate had a significant effect on evolution of new strains that had potential to spread globally, and they say a transmission rate of 17 to 28% higher in one region could explain historically observed patterns of flu epidemics, which bring it all back to what we've been talking about. We can see that there is what looks like a higher transmissibility, right? Which means that it's going to be very hard to shut this down. So when we talk about this in terms of being like a seasonal flu, I think we need to ignore the death rate of it for a second and say this is going to probably be with us. It'll be a lot easier once we get to the point where we have something to combat it, some sort of vaccine or some sort of immunity that gets built up within populations so that hopefully it won't be as deadly to as many people. However, if transmission rate is an indication of evolution, this one's probably going to evolve faster than the seasonal flu, meaning we may end up with several new strains of this over the years to come. So we just got to sort of be ready for knowing that that's a potential future here based on what these findings are saying. So understanding how and why seasonal flu tends to originate in these areas reinforces the need for surveillance to identify new strains. And this is also a very important part. This is not the Asia blaming for flus, but this is saying that a lot of the world's focus on handling pandemics should be located in that region. We should have perhaps a third party, if you will, that's making these sort of checks and observations and investigations into this area so that globally we can be more prepared. I think that there was a little bit of a problem in the delay of the rollout of explaining what was underway or revealing that there was something that needed greater attention. So I think some global dollars focused on preventing the next time would be well spent by all of us, obviously. You look
especially
at
considering the economy and what's going on with it now. Yeah, it's like we could have done a little better with. You put a little money in the preparation.
I think the
people
entire
are bad
world.
at that.
See, our country really likes to gamble. Is the problem? Is that
Not just our country, the world.
The whole world.
That's true.
But in our country, I will say that the toilet paper shortage that's going on in stores, this is the exact same reason that we didn't, as a country, as a world, we didn't put money into more research and preparation. And, there are things that we know are that the World Health Organization, that global leaders know are going to happen. People are writing about it. Researchers are working in the hot zones constantly. But yeah.
But
How do
this
you
is
manage
exactly
it?
what Justin's talking about. The fact that looking at this is not just the next couple weeks, but the next years or decades or beyond is really what this is about. Which is why there's been all this conversation about flattening the curve. Is if we can hold off. We know there's a certain number of people that are going to get infected, and it could be 100%. That is possible, right? And it could be a test of if we have a vaccine or not at that time, and how quickly it evolves and how quickly we develop antigens and all these things. But if we can make it not happen within the next 2 weeks, if we can stretch out when people are getting sick, then you won't overtax healthcare systems. You won't have dead zones in cities where services aren't available. And that's really what this is about, is slowing it down so that we can address this in a long term. Like, we're not going to get rid of it in 2 weeks. That's pretty clear. No. But we want to be able to handle it in the long term.
And something that we can agree or disagree about at some point, the numbers on this are all wrong. The fact that they're showing on that map that we had up earlier that the United States has 1,200 cases, we don't have tests to test for. I mean,
suspected.
You're having to work blind.
We're at, yeah.
These numbers are going to be largely reactionary at this point. All of them.
The fatality rate is going to shift based on what actually turns out to be people who have it versus people who don't. Because also a lot of those people getting the test kits are the most sick.
And as the flatten the curve idea is making its way through the public consciousness, the idea of it is to reduce the death rate because the healthcare system can't handle all the cases. If we can make that peak not happen over the next 2 weeks, right? If we can spread it out, same number of people get sick eventually, but if everyone can have a bed in the hospital who needs it, if everyone can have a ventilation machine who needs it, then fewer people will die as a result. So we have the ability to change the mortality rate by changing the curve. And we can do that through social distancing and other practices that, are going to affect the economy in the short term, which is true. Let's talk about fun things.
Yeah, let's talk about other science.
Let's talk about other science. If you tuned in, this is This Week in Science, and if you are interested in a fun shirt or mug or other item of TWIS merchandise, you can go to twis.org and click on the Zazzle link and browse our store. We have all sorts of cool items in there. Check it out. All right, Blair, I think you are going to be our guide into fun land.
Yes.
At least I hope you will.
So, yeah.
Is it time for fun time?
It's fun time.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's animal
What you got, Blair?
Who knows what biofluorescence means? It's the word of the day.
Biofluorescence, bio that fluoresces.
Yes. So it's not to be confused with bioluminescent, which bioluminescent things like bioluminescent algae creates its own light through chemical reaction. But biofluorescence is not a chemical reaction. It's something where an organism absorbs blue light and emits it as a different color. So it's kind of like it glows in UV light, like it's a black light image.
Yeah.
Cool. So guess who biofluoresces?
Me.
No.
Darn it.
A little bit.
I was really hoping.
A little bit.
That would be pretty cool, but I might have to ask you about your diet if that was the case. No, actually, it is most amphibians, we think now. exhibit biofluorescence. So this is a
Most
study.
of them.
Most. Well,
Wow.
that's asterisk. Hold on a second. So St. Cloud State University in Minnesota did this kind of far-reaching test on species of frogs, salamanders, newts, and eels, and exposed them to blue or ultraviolet light. And they found that many of them emitted this colorful pattern. They had either glowing blotches, glowing stripes, glowing bones, or even all over fluorescence in green, orange, yellow, or other colors. And some even had fluorescent green skin secretions and fluorescent urine.
Okay, so the fluorescent secretions in urine suggests that I could biofluoresce.
You could,
that
it would depend.
That
what
it is
you've
something
been
in their
eating.
diet.
Maybe. So, and it's probably not one size fits all here. But prior to this study, only one salamander and 3 frog species have been shown to fluoresce. But now that number is 32. 32 species, which is not all of the species, but it's a really good sampling. 32 species.
Yeah, I mean, what percentage of the amphibian species is this?
A
small percent, a big
100% of the ones that they tested.
Yeah.
So all 32 species they tested showed this. So that's why they think like probably most, if not all of them.
Right.
But further testing, I think, would be needed. But there's different mechanisms through which this is happening. So some amphibians also, in addition to whatever is giving them the fluorescent proteins, have chromatophores, so they can use that to adjust their coloring, or some of them also have light reflecting cells or pigment containing cells. So there's lots of kind of developed fancy coloration happening. Now in the picture we're showing, the upper right there is a tiger salamander, and this is an animal that I talk about all the time through my day job. And we talk all the time about how they have these bright colorations to actually warn potential predators that they are poisonous. Yeah. So depending on the animal and the size of the animal and their metabolism, it could give them a stomach ache, it could make them barf, it could temporarily paralyze them, it could kill them, depending how potent that poison is and the animal eating it. And so having this warning coloration, say, don't eat me, I taste bad, or I'll hurt you, is really important in the amphibian world. Think about poison dart frogs, right?
Yeah.
But so the thing here is that in all of the information that I read about this story, they talked about how biofluorescence could create a contrast between amphibians and their environment, allowing them to be detected for mating. It could help them to signal mates, could help them mimic the appearance of predators. But I think the most likely reason here is to make them really obvious and bright to go, don't eat me, I'm poisonous. Because a lot of amphibians are poisonous, and the ones that aren't, a lot of them mimic poisonous amphibians. So that seems like the obvious reason here, but I still think it's a pretty cool thing to know that you could stick most amphibians under UV light and they'd glow.
And also, if you think about the time of day that they're active, many amphibians, like you think about frogs that start, ribbiting in the evening or in the morning, right around that time of day when the insects come out, when the light and the UV light is going to be different in the spectrum. They're taking advantage of that in the light as well.
Yeah, absolutely. So the fact that most amphibians are crepuscular or nocturnal is a big part of that.
That's the word. Thank you.
So these are poisonous, but the poison doesn't kill the thing that tries to eat. It
can. It depends what the size of the thing is and the metabolism of the thing is that's eating them. So if a full-size crow ate one, it'd probably just barf.
Okay. Because that then would dissuade that crow from eating another one,
Yes.
and the markings would help.
Yes.
If it's the first time you tried to eat something and you die,
You're helping the
nobody
species as a
learns.
whole. That's what it's about. You're helping the species as a
Just
whole.
less
But
of
also,
you now.
we know
Eat something
that
else.
crows, for example, talk to other crows about what to eat and what not to eat. So bad news also travels fast. So that could be helpful there. I'll just also mention they found out that amphibian eyes do contain rod cells that are extra sensitive to green or blue light. So that's one of the reasons that they think that talking with each other is a really big part of this need. Ooh, a horned frog.
Yeah.
Yes.
Right.
Yeah,
so they can see it. But we know so much about also predator prey interactions that I would not be surprised if predators could see it too.
Yeah, totally. And as luck would have it, I have 2 biofluorescent stories in the animal corner this week.
Not one, but 2?
Not 1, but 2.
Wow.
The second is perhaps the animal most famous for glowing under UV light, and that is the scorpion. So, yeah, when I went to the Mojave Desert and we went for a night hike, we all had our UV flashlights on, looking for scorpions. We've known for a pretty long time, about 60 years, that scorpions have a fluorescent response to UV light. And until now, we only had 2 compounds that we kind of looked at the cause for that glow: beta carboline and 7-hydroxy-4-methylcormorin. You know, those. And they were in the outer shell or in their exoskeleton. But there's still this really big question of why, though? And no one's ever been very sure. But the previous speculation has been that it has something to do with sunscreen, or it helps them find each other, kind of like we were just talking about with the amphibians. But this is where it gets interesting. Researchers actually extracted compounds from molted exoskeletons of the scorpions that they caught, and they used chemical conditions different from those used in prior experiment to try to purify the compounds inside. And they purified the compound, showing the most intense fluorescence, and identified its structure. And it was a phthalate ester, previously shown to have antifungal and antiparasitic properties in other organisms. So the new theory is, why do scorpions glow? It's to ward off parasites. And they're not glowing to ward off parasites. It is a byproduct of the compound that wards off parasites.
Fascinating. An accident of nature.
Uh-huh.
Oh, I love it.
I accidentally glow under a black light.
Oops. Accidental glowage. I wonder, though, if anything has influenced aspects of their behavior, or, if there is some aspect of this fluorescence that has influenced their evolutionary path.
It's a good question, and
It's
I feel like
interesting.
for me to understand that this arachnid, this animal that has been around for so long, has this ability, but either before or after the fact has not found a way to use it to its advantage, seems insane to me. Because there's lots of animals that see UV. There's lots of UV light out there. It just seems like over hundreds of thousands of years of evolution, there would be a benefit. I don't know.
Yeah, that's what I would think. That there's got that, but who knows? Maybe there's nothing at all. Maybe it's just a strange quirk. And hey, there it is.
Who knows? Hey,
More
there
research.
it is. I have one last story about the coloration of animals, just to round it out, and it's about ship noise and crabs, but it's actually about what color they are.
What?
So I know. This is a University of Exeter study on shore crabs, which are the most common crabs on UK shores. They change color to match surroundings over long periods of time, over like weeks to months. And ship noise actually impacts their ability to do that. They don't depend on sound in their normal day-to-day life. So you wouldn't think, we know that noise pollution in the ocean affects other animals that use echolocation and other things like that, communication. But as far as we know, shore crabs don't use sound in communication or echolocation or any part of their day-to-day, as far as we know. But they put crabs in 3 different tanks. They expose them to either quiet underwater sounds, an hourly recording of ships passing, or an hourly loud occurrence of underwater sounds. And they were all dark shelled at the start and on white bottom tanks. And over 8 weeks, the crabs in the normal tanks with just normal quiet underwater sounds turned very light. But those exposed to ship noise only changed about half as much as in the other 2 tanks. So they did not camouflage as well. What's more, when they were exposed to a predator, which was an artificial bird, more than 85% of crabs exposed to quiet or loud natural sounds rushed for cover. But the crabs exposed to ship noise, it responded normally when there wasn't ship noise being played, but during noise episodes, were about half as quick in responding to the potential threat. So, A, sounds like they are using sound for something. But B, it sounds like this noise pollution is stressing them out so much that they can't devote energy to changing color. That has to be what's going on, if you ask me. But.
Yeah, it sounds like it's when somebody rakes on concrete or fingernails on the chalkboard, and then suddenly in that mode, there's people who react to it in such a way that they just can't do anything.
Yeah.
I discovered this about a friend of mine recently. I did not know this about them. There's somebody that works in brewing for as long as I've known them, just about, and somebody pulled a keg across the concrete and it made a little screech sound, and he just literally spazzed out.
Ooh, he's in the wrong business.
I know. How did you? This happens apparently not that frequently, of that noise, but yeah. So they, even if they aren't themselves using sound to communicate or do anything specifically with, or this noise does seem to be having an effect on them, and neurologically even. That's pretty wild.
Yeah, and let me read this comment from YouTube in what I assume is a very sarcastic tone, and I appreciate it. Wow, sound is a stress creator. Yeah, okay, yeah, we know. but for an animal that we don't think uses sound input in their day-to-day life, this is interesting to know. It's also interesting to know just for the real applications of this. If we know that noise pollution is affecting camouflaging animals,
yeah,
that's important for us to know for the general ecology of that species in that space. So it does mean we have to be looking in more places for the impacts of noise pollution.
Yeah, and we talk a lot about the impacts of noise pollution. So there's the idea of whales or other water mammals beaching themselves, potentially. The loud noises actually hurting them and doing physical damage. We also talk about it affecting reproduction in terms of mating habits, but I don't think we've ever talked about it in terms of stressing out the juveniles so much that they are unable to grow and reach their adult reproductive stage. Basically, it's keeping them in an unprotected or less protected juvenile state.
Yeah.
Yeah, they're not able to reach their full potential.
Yeah, it's a potential big problem, all just from some ships moving our stuff around. Bringing me my Amazon packages.
Get me the Amazon. Crabs. Sorry, crabs. I need my toilet paper. Yeah. All right. Well, we have done it for the first half of the show. I have a very, very quick message. We'll be back with uno momento in a moment for some more stories. I have some solar system news, and I think Justin has some genetics. We've got some things coming up. Stay tuned for more This Week in Science in just one moment.
things you've heard more than.
Oh, no. Sing it, Justin.
That's it. It looks like Portland has been put on full quarantine. They're not even allowing internet communications. No contact of any kind, even digital at this point.
I'm back.
You're back.
We're back. I'm back. Thank you. I'm back. I really own my whole computer. Thank you for listening to TWIS. You are the reason that we keep doing what we do every week. And we love to bring you up to date on science. What's going on in the world of science? What are the big conversations? What are the fun stories? What things are filling our head with curiosity and questions? Together, we can ponder these things. We can. And together, having these conversations, it is so much more fun. Additionally, we can bring a pretty sane perspective to a world full of misinformation and even downright fake news. So head over to twis.org right now, click on the Patreon link, and choose your level of support. Be a part of bringing sanity and science to more people.
And we're back with more This Week in Science.
Yes, we are. We're here with the science, and I hope that you're ready for the science. Justin, tell me a story.
Oh, so apparently 98% of our genome doesn't do anything.
What? It
Yeah.
does. Why would it just be sitting
It's
there?
just buffer.
No obvious function anyway. I guess it's only about 2% of the human genome that actually codes for proteins. And of course, proteins are the things that basically do all of the work in the human body. They do all the chemical reactions and the building and the structure of the things, and that's what we're made out of. And that's what makes us. Proteins everywhere. So genes are usually thought to be transcribed into RNAs off of the genome, which are subsequently then translated into proteins. This is central dogma of molecular biology. So what's the rest of this 98% genome doing? The entire assemblage, also they looked at the entire assemblage of RNA transcripts in the genome, the transcriptome. It contains RNA species that appear to have some other function different from coding proteins. They call these non-coding portions of our DNA because they don't make proteins. What are they there for? So this is a paper titled Gene Expression Networks in the Drosophila Genetic Reference Panel, which means, of course, that they did this discovery with flies. So this is many years of research. Clemson University geneticists Trudy McKay and Robert Anholt. They say their findings in here could significantly advance science's understanding of a number of genetic disorders. Because only, quoting voice, only in recent years, with the sequencing of the entire transcriptome complete, have we realized how many RNA species are actually present. So that raises the whole new question. If they aren't making proteins, workhorses of the cell, then what are they doing, says McKay, director of Clemson's University Center for Human Genetics. So what they kind of found was they identified more than 4,500 new transcripts within the Drosophila, the little fly, that have never been uncovered before. They referred to by researchers as the novel transcribed regions. These 4,500 transcripts consist primarily of non-coding RNAs that appear to be involved in regulating networks of genes that could contribute to normal function. They could also contribute to genetic disorders. So this is more Cody voice. Most disease-causing mutations are known to occur in the protein coding portion of the genome, known as the exome. But when you're only sequencing the exome, you miss the other disease-related factors and parts of the other parts of the genome, as such as the non-coding RNAs. So there's this whole section of our genome, the heterochromatin, which is apparently locked off. It's this really super tightly bound, wound up bit of DNA. Nothing gets transcribed and coded from it. It doesn't, like the rest of our genome, it doesn't get it doesn't get used. Apparently, though, they're finding that a lot of these RNAs are in there, and these RNAs are actually allowing for transcription of transposons. Transposons are these funny little sections that can pop into other places in the genome, sometimes disrupting what a protein might have been there. It can disrupt the formation of that protein if this little bit of floating DNA replicates itself in your genome in the wrong spot. You could end up with some sort of disease, an ailment, or a deficiency in ability to make a protein that normally your body needs. So this is a pretty significant discovery. They say non-coding RNAs may play an important role in opening up this region of the genome for expression of genes in a way that varies among individuals. So your own genetics could be determining which RNAs are able to go in there and open up this sort of lockbox of heterochromatin. It's again, I keep thinking that we've gotten further than this in our understanding of how the genome affects things. And I'm always reminded, no, we're still just scratching the surface on all of it. There's so much of this that is regulated then in what your gene expression actually is, which is why we can be 98% the same genome as a chimp, but we are expressed as very different than a chimpanzee is. When
60% the same as a banana.
Yeah, I mean, when you look at those numbers, you realize, okay, this isn't 1-to-1 on all, right?
That's just being alive. A banana's alive. I'm alive.
Yeah, but even but a lot of it was going to come down to then you have a basic genome, but that you share with all other living things. But how it's expressed, where it's utilized, how often something is utilized, all of these management factors are really what's determining how the code gets translated and actuated into a living, breathing creature. So it's very fascinating to know. I really did not know 98% of it was non-coding and somewhat considered somewhat mysterious in its function. That is
so. What you're saying is our genome is like the Marianas Trench.
It is.
You know what's on the
down
surface,
there?
but what's down there?
Yeah.
I wonder though, like, at what point, what technology is going to be needed to do real-time analysis of the changes or the expression of the RNA, the DNA, the RNA that's transcribed from the heterochromatin? How do we go in and actually watch while a cell needs to do something to see when it goes into its heterochromatin and opens it up? How do we do that? When do we do that? That's going to be amazing.
So we can actually do all of these things now. The equipment is not everywhere. I mean, we can look at time looks
like
and
specific moments in time.
Yes, we in real time is a crazy, wild. I don't know how.
Yeah.
We can do time points with the right equipment and the right research grant. You can actually go in and look at timestamps of when RNAs or maybe microRNAs are being expressed, and then you can correlate that with some function. But knowing that this supposed lockbox of non-coding DNA is actually getting coded off of from time to time is pretty wild.
It really is. Beyond even a locked box of things that we can't see and we don't know how they're getting transcribed, there are a lot of things in our solar system that we can't see, and we have had no idea that they're out there. Today, the our list of trans-Neptunian objects, whose orbits go out past Neptune, it increased by 10%. Well, not today, this last week. A research project looking at data from the Dark Energy Survey, of all things, has enabled us to see dark objects in our solar system, the far reaches of our solar system. These researchers used the Dark Energy Survey's data. It was high precision images of the southern sky. So only, not the whole sky, but the southern sky. A lot of the sky, though, that they looked at. This research, published in Astrophysical Journal Supplement series, describes the way in which they were able to take these images and started with some 7 billion dots of light in the southern sky, and then looked to see which ones seemed to move slightly over 6 nights of observation, at least. They got rid of stuff that stayed fairly static in the background. So they got rid of all the far-reaching galaxies and other stars and things that were in the background. And they got these 400 or so objects that they thought might be in our solar system. And the researcher in charge of the project, Bernardinelli, says, we have this list of candidates, and then we have to make sure that our candidates are actually real things. So they went back to the original data set to look for more images to see if they were around for, like, these objects were around for like 25 nights, to see if they could really get a look at these objects to see how they moved. So they ended up getting images about where an object should be. Became less of a dot and more of an object that would be in the night sky. They took multiple images, stacked them together to make them sharper and brighter in their imagery, in their reconstructions, and to confirm whether or not they were real. They verified their method was able to spot known trans-Neptunian objects in the area of the sky being studied, that they were able to identify fake objects that got injected into the analysis, so they could tell the difference between artifacts and real objects that are out there. After the ends of all of their painstaking analysis, they found 316 trans-Neptunian objects. 316 objects that were not known before, and brings our number up to around 3,000 objects that we know of out beyond Pluto. One of these objects, or multiple objects, this methodology could help us discover Planet 9.
Or debunk Planet 9.
Or debunk Planet 9, yes, by using this methodology. And they think they can go back and stack more data, use this method with other observatories, such as the Vera C. Rubin Observatory that's going to be coming online, and looking even further for more fainter objects in the distance. They'll be able to really confirm the objects that they've seen, be able to find new objects that are potentially even farther out than these objects. These objects, it's interesting. So Pluto is about 40 times farther away from the sun than Earth is. So 40 Earth distances from the sun. These objects go out from 30 times further than the Earth to 90 times further than the distance from the Earth to the sun, one astronomical unit. And 90 times out there is really far. And if we can see even fainter things, we can see even further away from our planet, away from our star, into the dark.
That's a mic drop if I've ever heard one.
Oh, yeah. It's pretty cool. I think it's just neat. It's, you think, oh, there's just, 8 planets now? But no, No.
Like 8.5.
Right. There are a lot of objects around our sun, and we still have so much to discover.
Yeah, that's what's crazy
So
to
much
me
is
is
out
we're
there.
not talking about way off around another sun. We're talking about our neighbors.
Yeah, it's our solar system.
Yeah.
It's won't you be my neighbor? Oh, you already are. Oh, okay, hi.
I feel like so much of the time when we talk about space, we're talking about stuff so much farther away
Mm-hmm.
that it's very easy to kind of get complacent and be like, well, we know what we have. No, we don't.
Nope. We have so much to learn. Justin, tell me another story.
Yeah, so this is sort of interesting. This was out of the University of Connecticut, but it's also University of Utah, Troy University, University of California, or California State University, Sacramento. Whole bunch of archaeologists and anthropologists apparently contributed to this. And they were looking at the growth of agriculture in eastern North America, 7,500 to 5,000 years ago. And they say they find that while the domestication of plants fostered new cooperation among people, it also saw the rise of organized intergroup violence, which is kind of interesting, I think, to me, just because this is also the same result that the rise of agriculture got in other parts of the world, and the four civilizations of the 3 rivers, 4 rivers. I can't remember how many rivers it was. One was in China, one was in the Nile, and then one was in like modern day Iraq. And when these civilizations rose up, very shortly after agriculture, they started fighting with each other. Now, one of the arguments has always been that as the populations got larger, they were more and more difficult to feed the people that were there. So then they would go and attack others to get resources. And one of the others is the groups that weren't being successful at farming would keep attacking the ones that had the resources so that they could feed their ever increasing populations. Although the one thing that I find that is interesting from this study, they have linked. So this is, let me go back to this. The transition from hunting from a hunting and gathering society to agricultural society is dependent on collaboration. This is out of Troy University, Stefan Carmody, quoting. The development of agriculture appears to only have happened in 9 places around the world. So Eastern North America is a unique part of the world to study. Agriculture was one of the most consequential transitions that happened in the past that changed our whole economic system. Developments such as combined efforts for harvesting, defense, and possibly even sharing seeds amongst different groups could happen with interpersonal cooperation, which then leads to greater chances of survival within groups. But there was this period that followed where there was trophy taking. There were signs of violence throughout history, but trophy taking was a very specific sort of violence that was taking place here, where they would, it sounds like a stereotype, but scalping of people, but also taking of ears and hands and different portions of your enemy that were being kept. And they say this was right on the heels of the agricultural revolution in North America. And then very quickly after that, there was a large dispersal of people people started moving further away from each other, which if trophy taking was becoming a thing, I think I would do too.
Yeah.
I think if
See ya.
I gotta go.
I see
I gotta go.
several hands on your farm that aren't doing any work. I don't want to be a farm hand there. So anyway, yeah. And as that happened, the trophy taking died down and eventually kind of was much less after people dispersed to further away. But what they point to is what they call a population density surpasses an optimum number, and then it declines in some point after. What's sort of interesting too is I've always seen this as one of the things that happens in certainly all of the river civilizations was that you would go from farming to this system where there were so many people that at some point there became a king, a leadership group that would take over and start managing. But the way they did it, these were farmers who had been running livestock, running agriculture. They begin running people like livestock, like agriculture. And so suddenly you have this taxation, which is not far from irrigation of funds into this central power. You have human slavery, which is cattle. It's actually referred to. Women were considered cattle at some point, written in some of these texts of ancient times. Everything was this, and everything was taken from the same thing that they'd been doing and just applying it now to humans.
It is interesting that in the narrative of ancient peoples, it almost kind of sounds like there were these hunters who were like bloodthirsty and violent. And then there were the gatherers and the farmers, and they were cooperative and they lived in peace. No, right. We're learning.
Yeah. So when you had, yeah, you're right. The hunter-gatherer groups were likely very cooperative. The women hunted and gathered right along with the men. We keep finding more and more these examples, so that there was a cooperative lifestyle versus when you start getting into these farming civilizations. Yeah, there was all of a sudden women's rights disappeared. It's like one of the first things to go, one of the first things that got taken away. And people were smaller and more sickly. That's another thing. Like the population grew, people got to eat more and grow, but they were smaller and sicklier than the hunter-gatherers were, because they were, getting diseases from communicable things, and they weren't
because they're around more people more often.
Yeah, and they're also
they're traveling greater distance. Yeah.
Yeah. And they're not running as much, chasing
Although,
the game.
I will tell you, there's a study out just this week also looking at modern hunter-gatherer societies, and they are just as sedentary as everybody else. So the whole idea of hunter-gatherers that we probably evolved from being so active all the time is not necessarily the case. If we look at modern hunter-gatherers as a representative of our ancestral.
So the transition period was interesting because at first they were just nomadic. People just followed game where it went. But then there became this awareness of seasons. Like if we go back to that one place where the strawberries grow at this time of year, and then cut across the valley, there'll be some bison or whatever running through. And then we can cut over to the ocean because that's when those turtles are up at the shore, or shells are. So they began to do tighter and tighter patterns. And then at some point, somebody noticed a bit of wheat growing in the trash pile, and we're like, that thing doesn't normally grow there. That's a way over. That's like 3 valleys over in a different place. What's it doing here? And then, oh, wow, you can actually bring those with you and plant them. And that's how farming happened and ruined the whole deal.
I know. I just got this whole image of humanity circling the drain to become society.
Oh, no. But you're right. For a lot of that hunter-gathering period, we would hit the same locations at least, and then narrow it down to like finding a good hunting ground and having a consistent effort over time in a smaller and smaller location. Which you're right, led to farming. But there was a time when we just were guessing.
Yeah,
that's how hominids ended up all over the continent of Eurasia. They were following one quick deer. They couldn't catch it. It took them from Africa all the way through to Siberia. One of these days. Oh, it's fast.
We'll get him yet.
That's right. Oh, speaking of one of these days old things, never mind. I have no good nuanced transition here. Things preserved in amber. There we go. We like things preserved in amber because they can stay preserved for a very, very, very long time. Researchers just published in Nature about a really, really exciting dinosaur find in amber. Now, when you think of dinosaurs as the ancestors of birds, what kind of images of those dinosaur bird ancestors do you have in your head?
Ooh.
So I obviously turned to Archaeopteryx pretty early in that conversation. So it looks like a always rainbow colored, even though we're not sure about that. Like toothed, vulture-y looking thing.
Tooth vulture looking thing. Probably how big? How big do you picture
Like
them?
the size of a medium-sized dog.
Right. You think of them as biggish. These, not little tiny things. Well, this little tiny dinosaur skull that they found encased in amber, in Burmese amber, it's the size of a modern day, the smallest hummingbird, the bee hummingbird. This bird skull that they found from the age of dinosaurs is amazing because it has a lot of traits that are very similar to modern birds, but yet also very similar to lizards and others. It had teeth. Like you talk about the toothed Archaeopteryx. This little tiny hummingbird-sized skull, it had teeth. And so this little... what it suggests, I mean, this little thing with teeth was going around snapping its toothy bill on its prey, tearing them up.
What did they eat? Worms?
Maybe they did eat little worm-like creatures. But additionally to letting us know that early birds, or the ancestors of birds, had really tiny forms and a diversity of sizes. They weren't just on the larger size. They were probably a lot of different sizes. It also suggests that there was a great diversity of all sorts of vertebrates at that point in time. And so amber continues to be a place where we can find secrets of the past.
Was it
Right.
pretty hot at this point in history? It was, right?
Yes. Yeah, it was.
So that's always something that gets me going too, is this conversation about whether these animals were warm-blooded or cold-blooded at this time, and what we know about surface area and how really big animals do best when it's cold. So they have less surface area per body weight, right? And smaller animals kind of do a better job of evaporative cooling, of adjusting their temperature if it's really hot. So knowing that, it makes some sense that they would be tiny, that there would be tiny animals for that reason, if for no other reason. You're thinking about the climate.
Right. Yeah, if you think about the climate, not all dinosaurs were giant. Especially at this point in time where a lot of evolution had taken place and birds were emerging. It was
It's pretty hard for me to buy that those giant dinosaurs were not in some way endothermic, because otherwise how did they lose heat when it was so hot out?
Panting.
Yeah.
We have to change all images of dinosaurs to have the big tongues hanging out.
But isn't panting something that generally is associated with endothermic animals anyway?
Right.
So
it is. It is.
Yeah, I don't know. It's kind of unrelated. Kind of related.
But a tiny bird that wasn't a bird. It was a dinosaur, and it had little tiny teeth, and it's cute, but also scary at the same time. And now I'm never going to picture a hummingbird the same, ever.
Oh my God. That's like, imagine having a hummingbird feeder on your porch, as I do.
And they've got teeth.
Instead of hummingbirds there, these little dinosaurs with teeth. Like, oh, they've returned. Hey, no, not my finger.
Yep. Gives you completely different appreciation for hummingbirds and dinosaurs. This is This Week in Science. If you want to help This Week in Science grow, get a friend to subscribe today. It's time for our quick science stories at the end of the show. Let's get down to some pillow talk, shall we?
Or
Pillow
is
talk.
this? You're gonna trick me. It's literally talking about pillows, isn't it?
No, actually,
Oh.
I am talking about communication after sexual activity.
Oh wow. Okay, yeah. I really thought that was a bait and switch.
Nope, not gonna do that to you this time
Oh
around.
my goodness. Let's hear it.
All right. So, what is the stereotype of the different genders when we're thinking about?
Girls want to talk and boys are already snoring.
Right. This is the stereotype. Exactly. And so you would think that if we were to study the effect of that communication, that communication would be like from the female's perspective, make them feel so much better about the relationship. It would calm down their stress response, maybe. It would, that you'd see a larger effect on the women than you would on the men. Well, some researchers published their article entitled Exploring the Effects of Pillow Talk on Relationship Satisfaction and Physiological Stress Responses to Couples' Difficult Conversations. They took 2 groups of people for 3 weeks, said, Group A, you will talk for double the time. You got to double your pillow talk after you engage. The other group, they didn't have to change anything that they were doing, and they came back into the lab, took saliva samples to measure their cortisol levels for stress responses. They asked them questions about their satisfaction in the relationship, and then they also had made them have difficult conversations in the lab, after which again they measured their cortisol to measure their stress. Well,
what were those difficult questions? I'm very curious.
I know. And what would it really be like to have difficult conversations with your partner in a lab? That wouldn't be difficult to begin with.
Let's talk about your mother. Why don't you talk about the last expensive thing one of you bought without the other's approval?
Yeah.
Do you think you help out well enough around the house?
Yeah, that's a good one.
Was that for me or for her? I don't. It was for me, wasn't it? Yeah, I knew it was for me. You know the answer. Why even bother asking? Of course not. Try. It's enough, isn't it?
Okay, so what ended up happening is that, surprise, surprise, they really didn't get much at all out of looking at the stress response. Pillow talk did not really affect anyone negatively or positively in the cortisol results. So
we don't need it anymore. It's perfect.
There are lots of reasons for that. So the results were not great in terms of they were insignificant with respect to the stress, but counter to what you would think, women did not have a greater relationship satisfaction from that double pillow talk, but men did.
Yes.
Men really liked that extra pillow talk.
Well, liked it or were more affected by it.
They were more positively affected by it.
Yes.
It made them more satisfied in their relationships.
Whether or not they would admit it.
More satisfied or more habituated to that contact, that continued contact, that reassurance. This is a really interesting timing of this because I was just reading about the forest baboon troop, which is this group of baboons that the more aggressive males left to go fight other aggressive baboons who were fighting over a garbage pile at some deep into the forest place where people were now living. And they went there, and then tuberculosis happened. All those aggressive baboons died. So this troop of normally very aggressive baboons lost their most aggressive males, and there was a lot more women or female baboons. I guess I don't know baboon.
Yeah, female. Yeah.
Female baboons.
Yeah.
Outnumbered, and sort of were made more in charge of the society. Now, the thing that happens with baboons is the male baboons leave their troop to go mate elsewhere, and new males come in from other troops. And what they discovered is over generations, as these young baboons who weren't part of this female society that was more female-dominated baboons troop that was more relaxed and less violent and had a lot more grooming. Males would even groom males, which never happened anywhere else. They come in with the old, how they were raised, and they were very quickly trained by the females with lots of grooming.
That's not how we do things here. Yeah.
With lots of grooming after sex. And they would habituate themselves to how that troop behaved and would start expressing within a very short period of time. They would convert to that troop's
the standard of behavior. Yeah, their
Yeah.
culture.
So this
That's
now I
cultural
understand what
transmission
the
there.
female talking after sex is. They don't need it for them personally. They need it to change the men and make them more part of their society. It's
Yes, I
Yes.
like it.
It makes
Yes.
perfect sense. Yeah. We're getting
Wow, I
trained.
never would have made that connection, but I think you're right. It just changes the way the men are looking at things, which is to the woman's benefit. I see. Interesting.
Yeah, very interesting. The study is interesting. It's available. We'll link to it on our show notes. My fast story, another one. This one published in Basic and Applied Social Psychology. Researchers look to find out whether or not someone being a hard worker or a genius was more motivating to people.
I know
And
the
they-
answer to that.
They compared how people responded to reading about Einstein or about Thomas Edison. Einstein being considered a genius, Thomas Edison being considered a genius who worked really hard to get what he got. And
Slightly communist,
that's the
but
idea.
okay.
And so overall, they did a number of different manipulations with students, and the idea was to look at how the idea of genius or hard work might impact the STEM, science, technology, engineering, and mathematics pipeline. Will students be more motivated to continue in the sciences by role models that are hard workers or role models that are geniuses? And it turns out geniuses do not help. It's those hard workers,
course
or
not.
at least, Thomas Edison was more motivating to students than Einstein.
Well, because if you're bad at, and I'm doing big air quotes here, bad at science in school, then you can think, well, I just don't have the natural ability. But if your role model is someone who worked hard, then anybody can work hard at a thing. There's a through line there. There's a very obvious rule there, which is keep trying. If you like it, you'll get better at it. But if it's all based on natural ability, then I'm out of luck if I don't have it.
Well, also, I think you would put yourself at great risk if you try to emulate a genius. Versus
I am
emulate someone who worked hard.
exceptional.
Yeah, if you just claim genius to begin with, which is why that fake story is really important. The fairy tale of Einstein being bad at math as a youth. It's totally made up. He was always really.
He was always a genius. He was always good, right?
Yeah. But that was a great story because then you get he was bad at math, but he kept trying and he became a genius at it through hard work and perseverance.
Well,
I mean,
also,
yes,
who
he
do
must
you
have
think?
worked very hard. Also, let's not undercut that, but still.
Who do you think was more well liked in their own time? A hard worker or a genius?
Ooh.
I
Interesting.
think I don't know. I feel like there's they're different kind of. When I first saw the headline for this story, I assumed it was about a workplace. Like, how do you respond if your boss is a genius or if your boss is a hard worker? And that's what it made me think about is what's the kind of person you'd rather hang out with? Who's more socially available? I guess
Obviously, I only hang out with geniuses.
Yeah. So
That's a good thing you're such a hard worker, Kiki. See, now both of them become insults. Like, you're a genius. Are you saying I don't work hard?
Yeah.
No, you're a hard worker. Are you saying I'm not really that bright?
I'm not smart enough.
Yeah, like, that's awful. You can't use either of them now.
Nope. Damned if you do, damned if you don't. All right, last story, Blair.
Yeah, you know who aren't geniuses?
Who?
People who feed monkeys.
Don't feed the monkeys.
Yeah. So
Jeez.
the moral of this story, before I even tell it, don't feed wildlife, and especially don't feed wild monkeys. It's a bad idea. This is a study from University of Lincoln in the UK. They tested people with photos of wild Barbary macaques, which is a primate that commonly goes up to tourists in North Africa. And they asked them to assess their faces according to a variety of traits, including dominance, trustworthiness, cuteness, and socialness. And then they were also asked to rate how close they would be willing to get to those monkeys to feed it or to take a photo. In the test, the people said they would be more willing to approach, feed, or take photos with macaques that they perceived as trustworthy, subordinate, cute, social, young, or female, which means people generally know which are the least risky choices to make. However,
However,
they gave me the
despite
actual
their
pictures.
stated preferences, yes,
Yeah.
in field observations, people, on the whole, tend to get closer to more dominant macaques, which are the most dangerous option. And that is because the dominant monkeys get closer to you. So your
and
odds
people
are
don't back off. Yeah.
no. And so your odds are, if you're in the wild and you're approaching a wild animal, the ones that will not have a large flight distance, the ones that are more likely to stick around as you get closer to them, are dominant and or aggressive.
Wait a sec. A macaque? Who wants to be anywhere near a macaque?
I know,
that's
it's
like
bonkers.
a violent, sharp, nasty type of monkey.
Yeah.
That's the worst kind of monkey to get close to.
I mean, I don't think there is a good choice in the monkey world, if I'm being honest, about what you would want to feed in the way of wild monkeys. But macaques are definitely a rough choice. But ultimately, this means there's two sides to this, and these people in these observations, it doesn't matter whether they knew it was a good idea or not. If the thing's right there, they're going for it. So please, people, stop feeding wild animals, especially monkeys. You're gonna get bit.
This should be easier.
A communicable disease.
I have to say, I think not feeding wild animals should be easier for people than not touching their faces. Just gonna put it out there.
Truth.
See which one is easier for you.
Good thing I'm at home because I've touched my face probably 100 times during the show.
I'm here and look at me touching my face right now while I do the show. Oh my goodness. Are we done?
I think we did it.
I think we did
done it.
We have. Thank you for listening. I hope you enjoyed the show. If you did, share it with a friend. It's time for some shout outs. Fada, thank you for your help in the chat room, for helping with social media. Gord, thanks for manning the main chat room. Identity 4, thank you for recording the show. And I would like to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Time now to thank our Patreon sponsors. Thank you to Paul Disney, Ed Dyer, Stu Pollock, Andrew Swanson, Craig Landon, Ed, Philip Shane, Ken Hayes, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Richard Porter, Mark Masaros, Bob Calder, Jack Matthew Litwin, Jason Roberts, Bill Kay, Eric Knapp, Richard Brian Condren, Dave Neighbor, Ashley Doyle, Maddie Perrin, Ben Bignell, Justin, Taylor P.S., Josiah Zahner, Howard Tan, Donald Mundis, Sarah Forfar, Dan K., Matt Bass, Darwin Hannon, Patrick Pecoraro, Jean Tellier, John Gridley, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney Lewis, Tiffany Boy, Mountain Sloth, Seth O'Gradney, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Noodles, Kevin Reardon, Christoph Zugnerek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds Flying, Brian Carrington, Christopher Dreyer, Lisa Slazuski, Jim Drapeau, Greg Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Gregg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for your support on Patreon. If you're interested in supporting us, you can find information at patreon.com/thisweekinscience. On next week's show, we are going to be talking about roving on the Mars surface. We're going to be talking with a mapping expert who maps planets and moons
That's
for JPL.
tight.
Yeah, NASA expert JPL scientist, works with rovers, the InSight mission, and we're very excited to be talking about space, the final frontier, next week.
So I guess Justin's not here, so I will continue. Oh, there he is. Okay.
Now he's
Where'd
back.
he go?
No, but go ahead. I'm still loading everything else. Everything glitched on me.
Okay, we'll be back next Wednesday at 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from TWIS.org/live. If you want to listen to us as a podcast, just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well. Maybe just pick a couple, grab their phones, add our show. They'll be pleasantly surprised.
That's right.
And for more information on anything you've heard here today, show notes and links to the stories we talked about will be available on our website. That's at www.twis.org. And you can also sign up for our newsletter there. Also, while you're there, or actually just anywhere with internet, you can contact us directly. You can go ahead and send an email to Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or what will happen, Justin?
You'll be spam filtered into oblivion. You can also ping us on Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.
I'm gonna hide behind our logo. Are you there, Blair? You there?
Huh?
What? Hello?
What?
Where are you? Where'd you go?
What?
That was funny.
I couldn't hear you from behind the logo.
Justin went away. Where'd he go?
I know. That's
Why
all
is he
right.
still there but not there? Like,
He waved. I think he's out for the count.
I think
Honestly,
he is.
I'm okay with that. I should go to sleep.
He's like, I gotta go. Gotta go. Yo,
Ed
he's
wanted
gotta
to know if
go,
I'm still going to classrooms, and that's one of the reasons I want to go to sleep. Is I want to make sure my immune system is healthy because yes, I am. Schools have not closed, and we have not had any specific directions to cease our normal procedures. So we're doing it because these poor kids are they're potentially going to spend a lot of time inside soon. So
Yeah.
I want to give them what I can.
Teach them not to feed the monkeys.
Yeah, I know. I actually did a suitcase for survival lesson today, which is pretty cool. That's when we have all of our confiscated animal trade products from Fish and Wildlife in a big suitcase, and we talk about wildlife trafficking and the difference between poaching and hunting and why wildlife trafficking impacts biodiversity, why certain wildlife trade is illegal, and what kids can do about it. So
Well, that's nice.
it's a pretty good one.
That is a good one.
Yeah, it's actually I feel almost
Interesting.
like it's a more effective lesson than it was when it started in the I think early 90s, because when you were telling kids in the early 90s, now don't buy animal products, like I don't think kids had as much agency as they do now, because now they have entire online social networks and can help with social norming, indicating that certain products are not cool to have.
Right.
Which is ultimately what we need, is to have demand be zero. Because as long as there's demand for these items, animals will continue to be poached. So if you can make it a huge faux pas to wear
Fur from
endangered animal fur or sea turtle leather or whatever it is. Oh yeah, we have sea turtle boots. Yeah, we have a sea turtle cowboy boot. Yeah. So if you can make it a fashion faux pas, then the entire reason to hunt those animals will be gone. So
yeah,
that's
really
cool.
What
it's about, but anyway.
As long as, like people can still have the things that came from, like, that they inherited from their grandparents or whatever, oh, I've got this ivory thing, whatever it is.
Totally. And so that's actually that's a cool scientific breakthrough that's happening more recently, is mass spectrometry, which I always say wrong, and DNA sequencing. These two things they can now do to animal products to help with the DNA sequencing identify the species, which is important because someone will say like, oh, that's not sea turtle boots, that's alligator boots. You're like, no, sea turtle, and I can prove it. But the other one is, you can actually, by measuring wavelengths and all this kind of stuff, you can identify if there's ivory that's a certain number of years old, or if it has just been scored and stained to look old. Because this is the issue, is that there's still ivory getting sold and passed as like as hand-me-downs, as like, as these older antique pieces that are actually brand new ivory, and people are scratching them up and staining them
making
and then
them
selling
look old.
them. So this has been a big issue, is trying to figure out how to cut through that. So there's science out there that potentially can help us, which is cool.
Help us. Justin, you came back. I thought that wave was I'm out of here.
Yeah,
we thought you left.
it was just I wasn't planning on coming back, but I gotta go.
That's fine. We gotta go too.
So this is like a real thing. We have there apparently is. I'm gonna Google to see who's initiated this, or maybe one of you can. We're no longer allowed to travel to Europe.
Correct. Trump did it today.
Today.
Yeah.
Like 2 hours ago. Hold on, let me.
So, yeah, I was only
That's not the cough into my hand.
I was only planning on being in the States for a few more weeks. So
That might work out.
It
might be lifted before the 30 days are up.
Oh, okay, I can look forward to that maybe.
But otherwise, it's an extra 9 days.
Yeah, but the thing is, like, planes, airlines. I mean, they have to, like, they don't just gonna be prepared for an if maybe, and then boom, it starts. This is really gonna be tough. So I'm in the States. I'm stuck here. If anybody's got a private plane heading to Europe, please let me know.
So it looks as though they're let's see. So according to foxnews.com,
a more reliable source we could not hope for, I suppose.
The measure will go into effect at midnight Friday. The strong but necessary restrictions would not apply to the United Kingdom. So you may be able to get to the UK and then take a train. Or a boat.
Wait a second. No, from the UK you can fly it anywhere.
Yes.
Yeah.
Oh, well, that's fine. That's where my connecting flight is. I'm good.
Didn't Denmark shut themselves down for 2 weeks?
Denmark shut down for 2 weeks.
Okay. Well,
so you gotta
in
wait.
2 weeks.
In 2 weeks.
You gotta wait a couple
All right, so this might be fine. All right. Well, that's a relief. At least,
so Europe is shut down, but not the UK.
Because the UK is no longer a part of Europe because Brexit.
But they're all connected by trains. This is...
I know. But I think
it's a bunch of islands and stuff. It's fine.
I think the point is, if you're from a European Union country, you can't fly to the U.S. If you're a U.S. citizen, it's not the EU. If it's not the EU who's initiated this, which is what I first thought, that this was an EU decision.
But they don't want people coming back, even if they are U.S. citizens, I think.
That's fine.
Yeah.
Thank you. Thank you. That's fine. It'll just be a one-way ticket. All right. I might be fine.
You might be fine. Yeah.
Okay.
Who knows? Okay. Who knows? We'll see.
All right.
Oh.
Oh my God.
I need to go to a better
hear
website.
The audio of this.
Right.
Stop.
Who needs to hear it?
Can you not autoplay? Internet. Message to the internet.
I can't wait until we live like in augmented reality all the time and like videos pop up in your visual view and it's like bloop-a-dee-boop-a-dee-doo, and you're like, I don't care.
Wait, that's how I feel about actual meat space reality as it is.
It's good.
Walking down the street, I'm like, I don't care. The sign, I don't care. The person talk, I don't want to hear it. The car going by, I don't need to hear your music. Like that's how I feel constantly anyway.
Yeah.
Yeah, it's so different. All right, I'm gonna go. I will see you.
Go.
I am interested in attempting something, a test at some point this week, because I've got I discovered that my phone's LTE hotspot on my bus works amazingly well for at least viewing, downloading, watching video.
You want to know about streaming from the bus?
I want to see if I could stream live from the bus, which is out in a remote farmland. It doesn't have like internet that I can tap into. But
right.
the LTE signal, they must have a tower out right near where this
Oh,
farm
yeah.
is.
So if you
because-
can use it as a hotspot and then
Mhm.
and stream. Yeah, possibly. I mean, I use
So-
a hotspot to stream occasionally. So if it's
Yeah.
strong enough.
Yeah. So I'd like to, at some point this week, we can figure out a time, whatever, we can text it out. I would like to do just a test run and see if, because that would be kind of exciting. You could
Yeah.
check out what's been going on in Busland.
That'd be fun.
That's very fun.
Yeah. So yeah. So let's huh?
You froze.
Yeah. We're still going. It's
Yeah.
So yeah. I'll text it out. Maybe we can one of these nights this week try to do a test run live from the bus and see if it's sufficient quality to try to do the show from there.
That'd be fun.
Yeah.
That would be fun.
Because
Yeah,
I
I'd
am.
love to do
This is
that.
also my stay here in the States for this couple of weeks is on the bus. I've moved into the bus.
Really?
I've got the hotel room
confused.
specifically to do the show, and also because I don't have proper showering facilities, so I'm going a little caveman
It's
for
nice
a large
to have
part
a
of the week.
shower.
You'd be mortified, Blair. Yeah, I'm going very.
True.
It's like an expedition.
I'm glad I don't have smell-o-vision on the
Yeah.
computer.
No, I'm fine now. I took a
This is his-
3-hour
he-
shower earlier today.
Yeah, the once weekly cleaning and TWIS
Yes.
episode.
Yes.
Kind of running together. Well, there'll be more than one of these visits a week, but yeah. So anyway, let's see if that can be a thing. That would be really cool. All right, I'm gonna go.
Okay.
See you next week, and maybe a little bit sooner.
Okay. Have a good one. Are we gonna go? Are you gonna go to
Sure.
bed, Blair? Okay. Do we have anything that we need to discuss before next week? I will get our interview set up for next week. We should do a newsletter and stay healthy, I guess. is the
Yes.
big
Everyone,
thing.
stay healthy wherever you are.
Wherever you are, wash your hands. Don't touch your face. Don't feed the monkeys.
Mhm.
Have a
Don't feed
cool-
the
good
monkeys.
advice. All good
Do we
advice.
still need to tell people not to feed the monkeys?
Yes.
Don't
Oh
feed
my
them.
I
goodness,
feel like
people.
there
Stop
was a
feeding
spike
monkeys.
in needing to tell people not to feed the monkeys once the Instagram was invented.
And I know this goes against a little what you're saying, but I just want to specifically don't feed the really aggressive, violent society monkeys. The ones that, if they have sharp teeth, it's for a reason. Stay away from macaques and baboons.
Hey, Justin. All monkeys have
I
sharp
know all monkeys, especially the one. Okay, you're right. I'll stay away from all the monkeys. We'll
And
see you
all
next week.
monkeys are very closely related to us, which means there's an opportunity for them to get you sick.
Mhm.
Oh, or the other way around.
Yes.
You could be killing all the monkeys.
Also true.
Yeah.
All right. All right.
Well, on
What
that
do you
note,
say
don't
now?
feed the monkeys. Say goodnight,
What do you-
Justin.
Good night, Justin. Say good night, Blair.
Good night, Blair.
Good night, Kiki.
Good night. Everyone have a wonderful week. Like we said, stay healthy. Don't feed the monkeys. Don't touch your face. I'm hitting the button now. It's over. No, it's not because I have